Okay.
Okay.
Cool.
So hello, everybody. It is my great pleasure to introduce our next speaker in the NGS Symposium series. Our next speaker is Máire Ní Leathlobhair, and she is an assistant professor in biological data analysis at Trinity College Dublin. Máire obtained her DPhil in 2019 from Cambridge University, where supervised by Elizabeth Murchison, she focused on understanding the origin and evolution of transmissible cancer. One of the results of Máire's doctoral research was a beautiful publication in Science discussing the evolutionary history of dogs in the Americas, and I really recommend you reading it if you haven't seen it yet. After her PhD, Máire moved to the University of Oxford, where she was a junior research fellow at Trinity College. During her time in Oxford, Máire was part of the cancer genomics group led by David Wedge at the Oxford Big Data Institute. And currently, her main research, I think, evolves around development of rare cancers, and we will hear more about transmissible cancers and how transmission cancer works from Máire right now. The floor is yours. Thank you.
Okay, cool. Great. So first I'd like to thank, I guess, the NGS team and Kasia in particular for giving me the chance to talk. I've been quite intimidated by the idea of online seminars and talks over the past year and a half, so I haven't given many. So I'm excited, but a little out of practice. Mostly this talk is a kind of rough catalogue of things that I've done during my PhD and since then. Some of the things that I want to do, and then just some things that I find quite interesting and I think are worth further exploration. So I guess what I wanted to do today is firstly preface this talk by briefly introducing myself and my research interests. And then I'll start by discussing some work I carried out about 3 years ago now in Liz Murchison's group in Cambridge. And then I'll follow up by discussing some more recent work still on the theme of cancer cell transmission, and then conclude with some of the work that I hope to do in the coming years while based in Dublin. So as Kasia just said, very recently I joined the Department of Microbiology in Trinity College Dublin, which is situated right in the heart of Dublin. I started out studying theoretical physics and then later maths here about 10 years ago before leaving for the UK. And at the moment I'm trying to build a program that bridges my interest in cancer and in microbiology. So broadly, the question motivating most of my research has been how genomic data can be used to understand clonal evolution and what this looks like in many different contexts. I guess starting with the very traditional view of cancer development, which is that cancer arises when normal cells are transformed into tumor cells by sequential mutations that allow them to defy normal cellular constraints and proliferate unchecked. So each cancer, probably already familiar with this, is the result of a unique evolutionary process as the cancer cells act to outcompete their neighbors and overcome host defenses. And this process is illustrated here, so in the kind of lower classical clonal evolution plot, with various clones, so I guess being represented by the white dots in an individual cancer gaining advantage and being positively selected for, driving waves of clonal expansion. So this is the expansion of these white dots, and then other clones being unsuccessful. And we know pretty well that this process often leads, unfortunately, to death of the host and therefore death of the cancer, which is an evolutionary dead end. But my PhD centered on a type of cancer that's gained the unusual ability to spread not only within the body, but also to spread between individuals within a species and then even between species. So these contagious cancers, they're not spread by a virus, but by actual physical transplantation of living cancer cells between unrelated individuals. So we might even think of this transmission as a sort of unprecedented next step in the invasion metastasis cascade. And obviously these rare contagious metastases have had to overcome very strong physical and immunological barriers by acquiring adaptations that allow them to transmit between allogeneic hosts across external environments. So this is sort of represented here in a human context, which doesn't happen very often. I'll touch on that at the very end of the talk. So in terms of transmissible cancers in animals, there are 10 naturally occurring transmissible cancer lineages or clones that have been described to date. So there's one in domestic dogs, there are 2 independent lineages in Tasmanian devils, and then several widespread independent lineages in marine bivalves, which have been discovered pretty recently, so during the course of my PhD. So each of these asexual lineages originated in some individual or founder whose somatic cells acquired these changes that first drove carcinogenesis, and then they continued to exist and proliferate long after the death of the individuals that first gave rise to them. So I won't be dwelling too much on Tasmanian devils or bivalves because that's not the area I work in, but I thought it would be interesting to mention a couple of cool things about those lineages. So Tasmanian devils are affected by 2 independent transmissible cancer cell lineages that arose almost 20 years apart. So the first was reported in 96 and another was reported in southeast Tasmania in 2014. And both of these diseases are transmitted by biting, which is why you see these quite gnarly facial tumors. And then the bivalve cancers are really quite interesting as well. So these kind of independently arising clones have been reported in soft shell clams. There are 2 independent lineages or clones in cockles, and also different species of mussels. So although the mechanism of transmission is not quite clear yet for the bivalves, it's been suggested that engraftment occurs during filtration of seawater that's been contaminated with transmissible leukemic cells. But in spite of how interesting these other lineages are, most of the work that I did during my PhD focused on canine transmissible venereal tumor, or CTBT for short, and it's probably how I'll refer to it for the rest of the talk. So it's a contagious cancer in dogs that's spread during mating. Unless you know an awful lot about how dogs mate, you might not know that mating in dogs involves a prolonged coital tie, which can quite often lead to injuries if the dogs try to kind of aggressively detach from one another, or sometimes if humans intervene and try to make that happen. So it's thought that the cells are physically transmitted during this process. And as a result, tumors usually manifest on the external genitalia of male and female dogs. So I thought it would be very often when I presented it's in these terms, kind of using nice graphical cartoons. But it's not always so sanitary. So in Liz's group, there's often opportunities to do field work and work quite closely with the veterinarians that are collecting samples. So these are some pictures I took when I was working in a clinic in Argentina, collecting some samples for analysis. So while CTBT is not quite so common in Western Europe, in certain parts of Central and South America, it's very common. So this was one patient to the clinic that came in wearing a quite sweet sort of tumor diaper. And I have some more gory pictures, but this gives you an idea of the kind of the extent of the tumor and its appearance. So the bulk of my PhD focused on, so what are genomic approaches that we can use to find out more about the clonal origin of this disease? So in the course of this work, I was able to confirm a couple of things. One of the more interesting aspects was the very ancient origin of CTBT, which had already been hypothesized. And there was already some evidence from genome sequencing data supporting this. But combining the genomic data that we had generated with ancient genomic data, and also using data that had been collected from a kind of a direct transmission that we were able to sample from, I was able to build a sort of molecular clock that confirmed that CTBT first emerged between 4 and just over 8,000 years ago, making this a very unique model of cancer evolution over the course of thousands of years. In fact, we think this might be one of the oldest mammalian somatic cell lineages. So part of this work is also comparing DNA from the CTBT founder with DNA from hundreds of modern dogs, as well as ancient DNA from dogs across the world. And one of the kind of more remarkable findings from this is that the dog that first contracted CTBT was most closely related to a group of ancient Native American dogs in North and South America. And at the time, we had sampled from one of the oldest archaeological dog samples. So these results showed that also that native dogs to the Americas weren't domesticated in North America, and also that this group of dogs is no longer present in the Americas today, having been largely replaced by European dogs over the centuries. But that actually the closest known vestige of these ancient dogs that have now disappeared turns out to be this weird tumor. And in fact, this cancer has possibly been affecting dogs for as long as dogs have been domesticated, and is sort of like a living relic of the distant past. And then in subsequent work, we looked at how the CTBT clone has spread around the world in the past few thousand years, and how it has evolved during this time, and used mitochondrial DNA as a way of mapping genetic diversity, which is not represented on this slide here. But using mitochondrial DNA as sort of a genetic tag, we're able to show how CTBT has spread around the world in the last 2,000 years. And then some other work that has been continuing, I guess, or has continued up until now, was developing screens or screening cancer outbreaks or pandemics in wildlife populations to determine whether any of these might be occurring due to clonal cell transmission. So essentially, screening or scanning the literature to find whether there are any reports of unusual disease prevalence in a population, or some sort of unusual distribution that you might not expect, and then testing tumor and host samples from these diseases to determine whether we're looking at kind of distinct tumor host genotypes, which would be a marker of potential clonal cancer transmission. So we ended up looking at first urogenital carcinoma in California sea lions, and then in a subsequent paper that was published in Wellcome Open Research this year, we had a look at goldfish schwannoma, and also green sea turtle fibropapillomatosis, which are all, well, in the case of the sea lions and the sea turtles, they'd be considered almost like cancer pandemics in those populations. In the goldfish schwannoma, it's really been a series of sort of anecdotal reports. So the samples came from a public fountain in Palermo, where there was a population of goldfish. And over the course of 3 years, it started with a report of a tumor in a single goldfish. And 2 years later, almost all of the goldfish there had tumors, the same sort of tumor apart from one. So we sampled from that fountain just to figure out what was going on there. But as it turns out, none of those are due to clonal transmission at all. So we're going to keep looking. But it kind of points towards how we might develop experimental approaches for monitoring cancers in wildlife populations. And then moving on from this again, so something that I've been doing recently is, well, in a very real sense, transmissible cancers have made a really unusual biological transition from a somatic mammalian cell to what is almost like an asexual obligate parasite. So essentially, they should be subject to the same core evolutionary processes as clonally reproducing unicellular organisms. The same could be said of normal cancers. But basically, the idea of some work that I recently carried out with Richard Lenski was how we might use or kind of translate population genetic theory as a framework that helps us think about microorganisms and experimental evolution, and apply that to transmissible cancers. So this figure is from a recent review article that we wrote together, which is sort of taking some of the more recent findings from transmissible cancer research, comparing these with predictions from population genetic theory for clonal diploids, as well as some of the findings from experimental evolution and genome scale studies of asexual organisms, and starting to think about also whether we might test some population genetic predictions about asexual populations in transmissible cancers, and in particular exploring some of the more interesting parallels between Richard's work on long-term evolution in E. coli and transmissible cancers, which are sort of like naturally occurring long-term evolution experiments. So this figure really is just to try to tie together some of those aspects of what happens in experimental evolution and what happens in transmissible cancers, in particular focusing on the idea of there being a serial bottleneck occurring as the cancers are transmitting between hosts, and what effect that might have on the genomes and the population genetics of these cancer cells. But obviously there are many ways in which cancer cell populations differ from microbial populations that are used in experimental evolution. And we also explore some of the ways in which these studies or populations might diverge. So in many cases, experimental organisms are usually haploid. For example, some bacteria and yeast that are typically used in these studies. And they evolve within controlled environments. So yeah, hopefully that will be out soon and make for an interesting read, and it's something we can develop some of the studies that we suggested there in the coming years. So another question that kind of fascinates me and is something I'm hoping to work on a little bit more is what transmissible cancer might look like in a human scenario, which I guess is a pretty self-centered way to look at all these interesting questions, but nonetheless, I think it's kind of fascinating. So in thinking about this, there are two immediate problems. One is that there's a very obvious barrier to migration of cells or physical transfer of cells between two individuals. And the second is the barrier that exists to invasion, so in the form of alloimmune recognition. So the first place to start to look in thinking about scenarios where cancer cell transmission might occur in humans is scenarios where one or more of these barriers is lifted. So this is from some very recent work that's still under review that I did while working with David Wedge in Oxford in collaboration with Adam Mead, a clinician based in Oxford, and one of his PhD students, Nicholas Sousos. So this case was identified by them, and we were able to join them in some of the kind of interesting genomics work. So basically what's shown here is a rare occurrence of in utero transfusion between twins. So it's a case of monozygotic twins that presented with CALOR mutation positive. So CALOR is the gene for calreticulin, and it's a quite frequently occurring driver in myeloproliferative neoplasms, which is what the twins presented with at the ages of 37 and 38. And what we found during this study was that in both twins, the CALOR mutation was absent in the T cells and dermal fibroblasts, which confirmed a somatic acquisition. And then by carrying out whole genome sequencing and lineage tracing, it revealed a common clonal origin of the CALOR mutant MPN clone, which in fact occurred in utero and was then followed by twin-to-twin transplacental transmission. So it's a case of very, very early developmental origin of CALOR mutant blood cancer presenting in adults as overt disease many decades later. And obviously this was a one-off case, but it could prompt quite interesting further studies. So for example, concordance of clonal hematopoiesis with identical somatic mutations has been reported in elderly twins. And this suggests that maybe clones that arise in utero might even persist lifelong until old age without causing disease. So how frequently this is occurring, I think, could be a really interesting question to explore. One of the other things that we found is that most of the kind of mutations we found after looking at this in utero origin were subclonal, so all at the level of subclonal drivers, say, but none of these appear to have had an effect on the resulting phenotype, as we're seeing disease present at roughly the same age and in roughly the same manner. Although there was a really interesting TET2 driver mutation in one of the twins, so I guess we can't rule out that maybe without this TET2, potentially they might have had different disease phenotypes. But as I say, it's a sort of one-off interesting clinical case study and opens up a lot more questions. So I thought it would be worth, given that I'm highlighting this study, looking at a couple of other recent interesting studies, both of which have appeared in NEJM. One appeared earlier this year. So this is the report of 2 cases of pediatric lung cancer. I think this was one case in a 23-month-old and another in a 6-year-old that actually come about as the result of mother-to-infant transmission of uterine cervical tumors. And this happened to be detected incidentally during routine next-generation sequencing of paired samples of tumor and normal tissue. So here it's quite likely that if the mother has cervical cancer, then potentially the infant can be exposed to tumor cells in fluids in the birth canal and could aspirate tumor cells into the lungs. And obviously transmission of maternal cancer is extremely rare. And in this paper, I think they cited just 18 previously reported cases. But it's still quite interesting that even in an incidental way where they weren't seeking to establish how often this occurs, they're able to find 2 cases. And it's quite likely that they will follow this up in a more deliberate way. But it's definitely a paper that's worth a read. And it's also probably worth mentioning another report from the New England Journal of Medicine, this time it was published in 2015, so you possibly have heard about it before. But it was a case where lymph node and lung biopsies in a man infected with HIV were in fact found to derive from a parasite, so a dwarf tapeworm. So even though tens of millions of people are thought to be carriers of dwarf tapeworms, obviously this is something quite rare. So the hypothesis in this pretty unusual case is that continued proliferation in the of the dwarf tapeworm in the immunosuppressed host allowed somatic mutations to accumulate in the stem cell population of the tapeworm, and this led to malignant transformation and subsequent invasion of human tissue and metastasis. But all of these cases sort of emphasize that cancer cell transmission can take place in situations where those barriers that I mentioned break down. So in an in utero scenario, or in a scenario like this where the host is immunocompromised. And following on from that, so part of the work, or part of what I've been doing over the past 3 years, or trying to do, and that I'm now initiating here in Dublin, is a study of another case, or another example of person-to-person cancer cell transmission. So pretty remarkably, a sort of close parallel to transmissible cancers can be found occurring in humans, and it's called gestational trophoblastic disease. So it's in fact a group of very rare pregnancy-related tumors that derive from abnormal placental development, which is, there's a spectrum of disease, so it ranges from benign manifestations. So here we're seeing a molar pregnancy. So in typical complete molar pregnancies, the placental tissue is abnormal and swollen and appears to form these fluid-filled cysts which is what's shown here. And in the case of these complete moles, which is just one form of the disease, there's no formation of fetal tissue. But in the case of a partial molar pregnancy, then there may be some normal placental tissue along with abnormally forming placental tissue. So I think the history of this disease in particular is quite interesting. This is a print from 1620, I think. As far as I'm aware, the author is unknown, but it comes from an account of the birthing, or I think the term is the accouchement, or the giving birth of Margaret, the Countess of Henneberg, on the Good Friday of the year 1276, when she reportedly delivered 365 children. So half of them were boys, baptized in the name of John, and then the other half were girls who'd be named Elizabeth. And in fact, in a church in the Netherlands, I'm not sure whether this is kind of legend or it still exists, but there's a monument for the dead children in the church, and also the vessel in which they had been baptized was still on display there. So this is part of what's represented in this print. And the vessel gradually became a pilgrimage destination for childless women who hoped to become fertile, and they would wash their hands in it. But the reason I'm mentioning this at all is that some consider this to be a very early example of a chorionic tumor, or one of these pregnancy-related tumors, where there would have been potentially, say, 365 of these fluid-filled sacs, although it's unclear whether that is really the case. So in explaining a little bit more about how these tumors come about and in what sense they are actually an allograft, so probably know already that trophoblasts are the building blocks of the developing placenta, and they're semi-allogeneic, so obviously they contain both maternal and paternal DNA. But in fact, in many of in the case of these molds, you might often see tissues that contain only paternal DNA. So it's an entirely aberrant type of fertilization. Normally it's described as a sperm fertilizing an anucleated egg, not containing any of the mother's genetic material. But as far as I'm aware, such an empty egg has never in fact been observed. So it's likely to be a different mechanism. But essentially, regardless of the mechanism, you have a scenario where only the paternal DNA proliferates. In some cases, as I mentioned, in some of the diseases, you will see the presence of some fetal tissue in scenarios where there is a combination of paternal and maternal DNA. But I think this raises a lot of obvious and interesting questions related to the work that I've presented so far, which is how is this happening? Is this potentially taking advantage of normal maternal-fetal kind of immune compromises to evade detection? And although it is quite rare, it occurs on a kind of a more frequent basis than some of these quite sporadic clinical case reports that I've mentioned so far. So there is a chance to establish studies looking at the biology of this pretty interesting disease. So part of what I've been doing for the past 3 years is collecting the samples, which is pretty tricky as it's a sensitive topic and a difficult tissue to collect. And again, it's a very rare disease. But hopefully in the coming years, I'll be able to explore exactly what is going on in this human instance of cancer cell transmission. So I was thinking it would take too long, but actually this brings me to the end of the presentation. So I hope I've kind of given you a flavor basically of the different phenomena or the different manifestations of cancer cell transmission. Sadly, the presentation is kind of pretty light on mechanism, but I think that largely reflects the amount of work that needs to be done in the field to describe exactly what's taking place in each of these instances. So yeah, if you have any questions or if you're interested in collaborating or visiting Dublin, yeah, let me know. And the hope is essentially that someday this work might have pretty important consequences in terms of how we understand immunology, cancer development, even reproductive biology, as well as some far-reaching translational possibilities from new drugs for transplant recipients to cancer immune therapies. But everyone has pretty big hopes for their research, I guess. So that's it, and happy to take any questions.
Thank you very much, Máire. I'm not sure if I can be heard when I'm talking to you, but I can be heard on the recording. Can you hear me?
Yes.
Okay. Shimon, can you hear me? Because I know that there was echo when I had my mic switched on Discord when I was talking to you on the phone, so I didn't want to repeat that. Okay, so my question, first of all, thank you very much for an amazing talk. It's really interesting and a little bit scary to hear about transmissible cancers, at least in my perspective. I had a question, and it might be a little bit weird.
Okay.
Is it possible to have a transmission of cells, of a cancer-derived cells, and then would it in theory be possible to infect someone with cancer-derived cells?
You mean if you were to implant cancer cells in? Yes. And so in fact, there are there's some additional case reports that I didn't mention, which would be considered iatrogenic. So sort of accidents taking place in a medical scenario where potentially a health worker would have been pricked and by a needle, or there would have been some transmission of cancer cells. So you do see some rare, again, sporadic reports of cancer cells transmitting in that way. There was also, although the name escapes me, a clinician who undertook a study like this, so enrolled, I think, death row prisoners in the US. As far as I'm aware, there's a big a piece covering this in Time magazine at some stage, but he sought to address this question exactly. So what if he took tumor cells and implanted them directly in individuals? Would they take? And I think in some cases they did. Broadly, there was an immune response. And obviously it's ethically kind of terrible thing to do, if not, it is really interesting. I did end up reading about it, but yeah, so people have looked into that. And it's very rare, but possible, which is sort of what a lot of the literature is telling us, I guess.
Very interesting.
Yes.
Do you think it's possible that we're going to see more cases of that? Like, not necessarily the cell lines, but
Yeah.
more transmissible.
I guess a good way to coax kind of funding is to say yes, and that we potentially need to be prepared for some sort of eventuality like this. There's a really interesting talk which Mel Greaves gave a couple of years ago at a conference at the Wellcome Sanger Institute. It's on YouTube. But he discusses a couple of the same cases that I've touched on today and a few more. And he kind of hypothesizes about different scenarios where this might take place. So I think one of the scenarios he proposed was, say, in individuals infected with HIV who might be already immune compromised and maybe also affected with Kaposi sarcoma, and that there might be some route for cancer cell transmission there. But without anyone sort of monitoring populations where instances like this might be taking place, it's difficult to say. So say in that report that I mentioned from this year, looking at the transmission between kind of mother and child from incidental reports. So once they go back and do a kind of more deliberate scan of the data, yeah, it would be interesting to see what comes out of that.
tumors in marine adult mollusks. Do you have a hypothesis why they are detected only in this organism and not in other marine invertebrates, which could also potentially be exposed to medicine in the sea?
That's a really interesting question. So no, at present I don't have a hypothesis. I guess mostly because CTBT, we've broadly known about it since, one could argue, maybe the late 1800s. Obviously, the DFTD, or the Tasmanian devil, the facial tumors, that's been since about the mid-90s. But really in the bivalves, people have only started looking in these populations pretty recently. So it's unclear whether other kind of neoplastic diseases in bivalves that appear at present not to be clonally transmitted might in fact be, because there were very few people working in the field of cancers that affect bivalves and looking at this question. So potentially it might be something that's quite ubiquitous, and then we might be asking, well, why are there so very few bivalves in which we see transmissible cancers? But overall, the answer is no. I'm not sure what the mechanism might be, but I will think about it.
Thank you very much. I have just one last question.
Yeah.
It's going to be more philosophical. Sorry, I just never really studied that. I just have more like, I'm super excited kind of question.
Okay.
What do you think we can get out of the concept of the transmissible cancers towards answering more questions about cancer progression, and how can we leverage it towards fighting this stuff? If you would draw parallels with this in the cancer research.
Yeah, I guess in terms of initiation and progression, the most interesting immediate question might be the basis of metastasis. So using it as a model to study what drives maybe kind of this sort of prolonged or super successful metastasis. For me personally, I think the most translatable aspect might be this whole idea of, how is it evading the immune system? Understanding that could be really useful for a couple of different applications. And potentially in each of these different clones or lineages, they might have evolved very different mechanisms of immune evasion, especially considering how different the kind of immune system is in bivalves, say, versus other species in which you see transmissible cancers. So kind of what is going on there. And in humans as well, looking at each of the different routes by which cancer cells are transmitted, how immune evasion might be happening in those scenarios. But it would be interesting to explore in a more philosophical way, like maybe writing up a set of just hypotheses and thinking about it outside of the data. But I guess that isn't too popular.
I mean, when you were talking, I don't know if it's a case of like, I'm a hammer and I'm looking for a nail, but with what I'm thinking about how chromatin organization, flexibility allows for better cancer survival strategy and potentially metastasis and such as endometriosis. I was thinking that maybe there's something to it, but I guess it's still, it might be a case of like hammer and a nail. Like the way I'm thinking, I would like to see that chromatin plays a role everywhere.
No, I think that would be it would be so interesting, especially when I guess writing up papers during my PhD and beyond. Largely, those efforts are again based on whole genome and exome sequencing, where you're sort of saying, well, we can find, it just seems like you're touching on a very small part of what might be the mechanism driving these tumors. And as far as I'm aware, I haven't seen any chromatin data from any of the the transmissible cancers in animals. So it would be definitely cool to have a look at that.
I'm just going to pass on to you some more things and comments from the audience. It was a really interesting talk. I'm sorry for the technical difficulties. I hope we might do this next year in person, hopefully.
Cool.
Then we would avoid all the connection issues. But thank you again, Máire. This was an amazing talk, and I'm looking forward to more and more research you're going to do.
Yeah.
In this area.
Cool. Thank you for listening and for having me. Okay. Cool.
Bye-bye.
Bye.